Replication, in the context of genetics and molecular biology, refers to the process by which a cell duplicates its DNA to produce an identical copy. This process is essential for cell division and the inheritance of genetic information from one generation to the next. DNA replication occurs in both prokaryotic and eukaryotic cells, but I'll focus on the process in prokaryotes. Prokaryotes are single-celled organisms that lack a true nucleus and membrane-bound organelles. DNA replication in prokaryotes is a complex and highly coordinated process that involves several components and steps. Here's a detailed description of DNA replication in prokaryotes. Components of DNA replication. DNA template, the original DNA strand that serves as a template for the synthesis of a new complementary strand. DNA polymerase, enzymes responsible for catalyzing the addition of nucleotides to the growing DNA strand. In prokaryotes, the primary polymerase involved is DNA polymerase III, while DNA polymerase I is involved in repair and completion of Okazaki fragments. Primer, a short RNA or DNA strand that provides a starting point for DNA synthesis. Primase is the enzyme responsible for synthesizing RNA primers on the template strand. Nucleotides, the building blocks of DNA, consisting of adenine, A, thymine, T, cytosine, C, and guanine, G. Complementary nucleotides are added to the growing strand during replication. Helicase, an enzyme that unwinds and separates the DNA double helix by breaking hydrogen bonds between the base pairs. Single-strand binding proteins, these proteins stabilize the single-stranded DNA regions that are exposed as the helix unwinds, preventing them from reforming double-stranded regions. Toposomerases, enzymes that relieve the torsional strain created by the unwinding of the DNA helix. They break and rejoin DNA strands to prevent supercoiling. Ligase, an enzyme that seals the gaps between the Okazaki fragments on the lagging strand by catalyzing the formation of phosphodiester bonds. Steps of DNA replication in prokaryotes. Initiation. Replication starts at a specific site called the origin of replication, or E. coli. The origin contains DNA sequences recognized by initiator proteins, DnaA, which bind and initiate unwinding of the DNA. Helicase unwinding. Helicase enzymes, aided by DnaA proteins, unwind the DNA at the origin, creating two single-stranded DNA templates. Priming. Primase synthesizes short RNA primers complementary to the single-stranded DNA templates. DNA polymerization. DNA polymerase III adds nucleotides complementary to the template strand in the 5-3 direction. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously in small fragments called Okazaki fragments. Proofreading and repair. DNA polymerase III includes a proofreading function that checks for errors in base pairing and corrects them. Mismatched nucleotides are removed and replaced by exonuclease activity. Ligase sealing. DNA polymerase I removes RNA primers and fills the gaps with DNA nucleotides. Ligase seals the nicks between adjacent Okazaki fragments. Termination. Replication reaches the termination site, and the process is completed. In prokaryotes, the termination site, there, contains specific sequences that trigger the termination of replication forks. In summary, DNA replication in prokaryotes is a tightly controlled and precise process that involves the coordination of various enzymes and proteins to ensure the accurate duplication of genetic information.